Good morning, students. Here is the chapter number 15, Plant Growth and Development. Following are the contents of this chapter: growth, differentiation, dedifferentiation and redifferentiation, development, plant growth regulators, photoperiodism, vernalization. Out of these topics, we will be studying plant growth regulators in more detail. Let us have a look on the remaining topics in brief. Here is a diagram that shows a growth of a seed into a plant. Whenever the seed germinates and the seedling develops, it results into a fully grown plant. We can see soil in which the seed is developing. Soil acts as the reservoir of the nutrients to every developing plant. The cotyledons start nourishing and the hypocotyle give rise to the root system. The epicotyle give rise to the shoot system. Seed coat gets removed, after which and cotyledons also shed off. Once the plant is having enough amount of leaves available for photosynthesis. So this is how a seed grows and develops into a seedling, and how this seedling grows and develops into a plant. How can growth be measured? So it can be measured in terms of height of the plant, root system, shoot system, or number of leaves present on it, or the girth of the stem. So there are so many ways. We cannot measure the growth directly, but we can measure the growth of the plant parts and can relate it with the overall growth of the plant. So this way, how we can measure the growth is, it can be measured in terms of number of cells and then relate to the whole plant. Phases of growth. So period of growth is generally divided into three phases. First, meristematic, which is the actively dividing phase. Elongation, in which the length of the shoot and root gets elongated. And maturation, in which the plant starts maturing its parts and starts the secondary growth. There are various growth patterns. We can see here in this diagrammatic representation, arithmetic growth pattern, in which only one cell continues to divide and one cell gets fixed. Second is the geometric pattern, in which all the cells divide equally. And third is the arithmetic with the geographic stages during the embryo development, in which both are occurring simultaneously. Dark blue colored cells are capable of dividing, while light blue are fixed. In geometric growth, we can see a large number of cells very soon. In arithmetic growth, we can see the growth is in limited way. But the third one, that shows both, first of all, the zygote divide into two cells, then four, and then out of the four cells, only some are capable of dividing continuously and some lose the capacity to divide, and an embryo is developed. There are three terms: differentiation, dedifferentiation and redifferentiation, very oftenly used in the plant growth and when we study growth. Differentiation means when the cells are assigned their own work. If the cells are given their function and they start doing that function in a very special way, then the cells are called differentiated cells. Then dedifferentiation, if the differentiated cells again start dividing, neglecting their function, then the process is known as dedifferentiation. The dedifferentiated cells divides and then again start differentiation. Since this is the second time they are differentiating, they are known as to be redifferentiated. So this way, these three terms, differentiation, dedifferentiation and redifferentiation, plays a very important role in plant growth and development. Here is the sequence of developmental process in a plant cell. We can see that the meristematic cells continues to divide. They show plasmatic growth, that is increase in the cytoplasm. Then differentiation, they differentiate and they start their particular function. After which some of the cells do not differentiate, they are meant for the expansion or elongation. They become mature. After differentiation and maturation, whatever mature cells are there, they will live their life, and after a certain stage or after a certain time period, they will undergo senescence, that is the old age, aging in the plants, in which the growth is negative. The cells that are lost are repaired or regained in a very slow speed, and ultimately they leads to the death. So here how cell division results in the meristematic cells. All the cells which divide are always in the meristematic phase. From that they have two choices: to continue divide or to fix and go towards the maturation. So those who continues to divide are always considered as meristematic cells, while some which are getting mature day by day are elongation and maturation cells. So this was the sequence of the development process in a plant cell. In the next lecture, we will be studying about the plant growth regulators, because plant growth is a very regulated mechanism occurring in the plants, and for that there are some regulators which are responsible for the growth. They trigger the growth and they inhibit the growth timely as the requirement of the cell. We will study that in the next lecture.